let's learn about your body senses, right? So last week we talked about sort of two sensory systems, right? We discussed taste and we discussed smell, right? Gustation and olfaction. We lumped those together, right? We called those the chemical senses, right? And we said that's all, sort of one thing together. Today we're going to take a similar approach, right? We're going to talk about various senses at one time. We're going to call this the body senses. Okay. So we're going to talk about several sort of sensory systems or modalities at once, but we're going to lump these together. Once we get past the body senses, right, we're going to spend a good bit of time just on hearing, and then we're going to spend most of the semester on vision, right? And so again, vision being one of the more important senses to humans, we're going to spend a lot of time on that. So we're going to kind of take a slightly different approach for the next couple sensory systems, and we're going to talk about those sort of independently. All right, there's supposed to be a picture of a guy doing a handspring. We're just going to draw him. I know, this is great, isn't it? Okay, so there's a guy doing a handspring. I know, it's art major right there. So a guy's going to do a handspring, which is kind of interesting. Why do we kind of want to start with that? Because we're going to think about proprioceptive information we're going to get from our bodies about our body position, right? Where we are, what we're doing. Most of you are aware of where your bodies are. If you're not, please don't tell me, because that seems like something I'm going to have to report to somebody. I just don't want to do that. So most of you are aware of where your body is without looking at it, right? You know where your legs are. I've seen most people walk. You don't have to look down at your feet the whole time, right? There are some strange situations where you would have to do that. Some folks have some brain damage or some disorders. So let's think about the different sort of body senses sort of in general. One of those we're going to think about is called touch, right? This is sort of how we're going to interact with that world in a physical way. Sometimes you'll hear this referred to as like haptic sensation, right? Any of you ever played those games where you got to like stick your hand in a box or a bag and try to figure out what's in there without looking at it, right? Okay, so there we go, right? So it's not the weirdest thing you've ever heard of, but it happens. Some people are pretty good at that and some people aren't, right? So that's one of the senses. When we think about touch, we're going to think about things like texture, warmth, softness, other sort of features. We have something called proprioception, right? This is going to let us know where your body is located, right? The position of parts of your body. This is sort of static information, right? This is like, where are you when you're sort of at rest? You have kinesthesis. This is when you're moving, right? And you can imagine that's dynamic information that's coming at you quickly, right? As you're moving through the world. You want me to draw that handspring again? You guys can think about how people move through the world, right? And you're getting that information coming back to you. So those together with the vestibular system, and we'll talk about the vestibular system a bit later. This is going to be like your balance and information about that. What's really cool is you actually have a sensory system dedicated to telling you which way is down. In case I know, right? You think like that's going to be something that's not going to throw you off, like which way's down. But you have a whole sensory system dedicated to that, which is going to be pretty exciting. All right. When we're thinking about things in the what we might call our cutaneous senses, right? Here's kind of another word. These are things that are going to be embedded in the skin, right? So these are things we're thinking about: touch, pain receptors, hot and cold, right? These are things that are going to be embedded in the skin. There are certain ways that these are structured. You know, in the first week we talked about some basic organizational principles when it comes to where sensory cells are located and how they're structured, right? And so if you start to think about the different kinds of information you're collecting through these cutaneous senses, you can think about the different way these receptors need to be structured and where they need to be located, right? So if it's something that's going to be kind of light touch, if you want to think about what's an important thing, maybe not for you today, although it's still quite important, you just don't think about it. A really important thing for our species in the past, and again still today, is determining if there are parasites crawling on your body, right? How many, I know, right? That's the face you make, right? How many of you have ever had a parasite on your body? Everybody just raise your hand, right? Everybody's had one, just it happens. You walk around, no, never happened to you, right? You've never at all in your entire life have you had a bug crawl up your arm, right? Or right into your ear. I had a moth get in my ear one time. That was a rough experience until I could make it to the bathroom. Usually the bathroom is a good place to go. You can turn out all the lights. Most bathrooms don't have a lot of windows, although on some level you might want your bathroom to have windows. It sort of depends on, you know, what your experience in there is. But they don't typically have a lot of windows. You can turn off all the lights and then use like a flashlight to try to attract the moth out of your ear. It's usually the best approach, right? If you try to go to your kitchen and your kitchen has a bunch of windows, it's not going to really work as well, right? Okay. So we have these cutaneous senses, superficial receptors, light touch, right? You want to see if there's like a parasite crawling on your skin. You definitely want to remove that. But you also have these deeper receptors that are more responsive to things like pressure, right? If somebody steps on your foot, right? That's a different sort of sensory experience than a fly landing on your arm. You need to have a different receptor sensitivity for that, right? So you're going to have a different thing. If every time somebody stepped on your foot it felt like a fly was landing on you, then you can see where you just have smashed toes all the time, right? Because you would never realize like that's a problem. On the other hand, if every time a fly landed on your skin it felt as though you know, your arm was getting smashed with a steamroller, you can see where that would be that wouldn't be helpful either, right? So you kind of have this balance. Here's a nice table kind of breaking down. Again, we've seen something very, very similar to this before when we talked about the location and the structure of those receptors. Importantly, with touch, this is going to be what's really going to determine what's going on. I'm not going to ask you to memorize all of these different names, right? This can get pretty complicated. If I go, hey, tell me about those you know, Pacini's corpuscles, right? I'm not going to ask you to memorize that. What I want you to think about is that relationship between the information that's coming in and how we might structure that receptor to suit that, right? And so think about that as more of a global level. It'll help you with this. It'll help you with other things as well. Okay? So those are going to be touch. Proprioception, similarly, we have stuff that are receptors in the muscles, the tendons, and the joints, right? Those are sort of the three big places that are going to be moving or going to be giving you information about your body position, right? Think about the stretch of your muscle. Think about the angle of that joint. If we have receptors in those locations, we can get a lot of information about that, okay? Right. There are sort of two ways that somatosensory information, and soma means body. So there's sort of two ways that this body information gets into your brain, right? Collecting all this information out in your arms and legs and fingers and toes is not going to do any good if you can't get it into your brain so you can think about how things go. There are two pathways. One of them is the spinothalamic and the other is the lemniscal pathway. I'm not going to get too upset if you don't remember the names of these two pathways, right? But I want you to know that there are two pathways and that information goes in, different information travels in each of these pathways, right? So for example, in that spinothalamic, we have what we would call sluggish pain or kind of slow pain and temperature. This is information coming from those free nerve endings, right? Okay. The other pathway is going to be faster information coming in from the mechanoreceptors. These are receptors that are sensitive to actual, you know, physical manipulation. Okay. Questions about that? Great. The somatosensory pathways can be a little confusing, right? It can be something that is, there's a lot of information that doesn't go all the way up before it goes somewhere important for processing, right? So there's ascending information, again, we have that spinothalamic and that lemniscal pathway. They go to slightly different places in the brain or in the spinal cord. Some of that's because you have to have reflexes. Okay. How many of you have ever been to a medical professional where they karate chopped your knee, right? And then you kick them in the face, right? That's a reflex. That information never makes it up to your brain, right? There's no way to really get around that. Have you ever tried not to do that? Right? You're like, watch this. I'm going to make this guy think I've got a neurological disorder. Hold my leg as tight as I can, and it still moves, right? There's no real way around some of these reflexes. The reason is it never makes it up to the brain, right? Once you get into the brain, you go to a region called the thalamus. This is that story I told you that happens for every sensory system except the first two we talked about, right? And I said, okay, so I've already sort of told you a lie, but here we go. From there, we go to what's called the primary somatosensory cortex. This is the primary receiving area in your brain of information about what's going on in your body. This is organized in a really interesting way, and I'd like to take some time to think about how that somatosensory cortex is organized for a couple reasons. One, it's really interesting. Two, it's going to be on the exam. And then the third thing is, once we understand how one sensory system is organized in your brain, we can just make minor tweaks and think about how other sensory systems are organized as well, right? And so that can give us that sort of additional boost when we go and we talk about other sensory systems. I can say, hey, remember that time I told you about how it was organized according to the structure of your body? Well, now it's organized in relation to the map of the world, or it's organized according to different frequencies, right? So we can think about that organization in a slightly different way. So for thinking about your sensory cortex in general, right? Your cerebral cortex is that wrinkly outer part of your brain. Looks like, kind of chewed up juicy fruit, right? If you were to like look at the brain, it's exactly what it looks like, right? Do they still make juicy fruit? Is that still a gum that is out in the world? Okay, well, there you go. This bit of cortex is sort of located, like if you think about where your ear is, it just kind of goes up, right? You have the same on the other side. You've got one on each side, one strip of cortex on each side of your brain that's responsible for receiving incoming signals. Now, the interesting thing about this is the strip of cortex on the left side of your body, stage left, I guess was over here, is the same, is getting information from the right side of your body. So it's a contralateral sort of situation, right? So the left side of your brain receives information about the right side of your body. Right side of your brain receives information about the left side of your body. There are some caveats to that we'll discuss later, but it's not too big of a deal. Now, once you get into the cortex, right, we say the cortex has 6 layers of cells, okay? The reason we say that is because like 100 years ago, when somebody first started looking at your cortex under the microscope, they were like, huh, this thing's got 6 layers. Well, it wasn't too long after that someone came up with a better microscope and a better way to visualize cells in your brain, right? And so then they realized, oh man, you know that time I told you it was 6 layers, and I've been doing that for like 5 decades. In fact, there are some other layers in there, and maybe sometimes it's more like 10 layers, and sometimes it's not. So what are we going to do? We've already told everybody it's 6. Well, we'll leave it at 6, and then we have sublayers, right? And so sometimes you'll hear about like layer 4, for example. This is a big layer because this is the layer of your cortex receiving all of that incoming information, right? And so there's like layers 4, and layer 4 breaks down into, like layer 4A and 4B and 4C, right? And so that has these sublayers. I don't want you to get too bogged down with that. If you sort of keep this 6-layer framework in mind, it works most of the time. It works well enough, right? It works well enough for you to get by, okay? If anybody's really interested, your cortex is sort of organized as a column, right, of information. And I know that's kind of hard to think about with your brain's kind of curved, right? But if you think about these perpendicular columns, what's really cool about that is if you go down through one column, The way this is organized is that like all of the like everything in that column is going to be sensitive to the same kind of sensory stimulus, right? Whatever that is, okay? What's cool is you can take a bunch of these columns and put them together into what they call a hypercolumn, right? And in that chunk of a hypercolumn, you can have all of the sort of sensory receptor types that you need, right? All of the information that you're going to be getting from your body. So if you think about one column collecting pain information, well, then you need another column next to that for temperature, and you need another column next to that for tickle, and one for itch, and one for texture, right? And so you can kind of put all these columns together, and you can organize those in a way that you have these just repeated hypercolumns, right, that represent different parts of your body. Okay, we're going to think about a moment in something called a receptive field. Right? This is a bit of a complex, abstract thing for you to think about. This receptive field concept is going to become particularly important when we talk about the visual system, right? But we want to introduce it now. Okay, so there we go. We've got your 6-layer business. The next thing we want to think about is a concept called cortical magnification. Okay, so I want you to think for a moment about, I'm going to ask you to make a map, right, in your head. Okay, if you were to make a map of, let's see, I'm going to assume most of you live in the Huntington area, right? I think that's a fair assumption. We can kind of draw like a broader circle maybe to spread out just a tad from that. But for those of you that don't, this will work just as well. I want you to think about drawing a map, okay? And of that map, I want you to include campus. I think you spend a good bit of time here on campus, right, Aspen? I'm just going to make that assumption. And then I want you to think about your residence, wherever that is. And I don't want you to think necessarily about, like, you know, where your mom and dad live if you live somewhere else, right? Like where you live, right? Like where you spend most of your days, okay? Because again, I'm going to assume you spend a good bit of time at that place as well, right? And if you were to draw that map, and that map's got to include both of those locations, right? Now, however far apart they are, it doesn't matter. There are going to be certain parts of that map that you're going to be able to give me more detail, right? And certain parts of that map that are going to be rather devoid of detail, right? Because they're not going to be as important to you. You're not going to spend as much time there, right? So let's imagine, anybody live in Beckley? Yeah, I knew somebody's from Beckley, right? There's always somebody from Beckley. I just assumed that. And some of you are going to, I always make this joke, but Andrew, this is going to happen. When it comes to exam time, and it's not going to be you, one of the rest of you, at least one, is going to have to go to Beckley for some reason. Like, what is there to do in Beckley? Exactly. Like, you listed everything just right in that inward breath listed everything you can do in Beckley, right? That's it. That's it. But for some reason, at least one of you will have to go to Beckley on exam day. So please don't use Beckley as an excuse. I know it's fake. I'm not even sure that exists. You live in Beckley. You could probably draw me a map of the part of Beckley around your home, right? Okay, with some detail. You can draw a map of campus in some detail, right? But if you were to draw the space between campus and Beckley, how much detail is going to be in there? Probably not as much, right? Exactly, that much detail right there. Because you're driving, what, 60, 70, 90 miles an hour, right, between there and here. I just assume. That's how fast I drive, so I'm assuming you drive that fast, right? You've got less skill at driving and less intelligence than I do, so you probably even drive faster. So you're going to drive, there's not going to be any detail there, right? Because why? That part's not important, right? What's important? Campus and your home, right? And then maybe you spend some time somewhere else and you can have some detail there, right? The whole space in which you sort of move about is not equally important, right? Some of it's more important than others. Your brain's the same way, okay? You've got a limited amount of space, right? How big is your head? Okay, now I want you to slap off your face, okay? Chop off your neck, right? Peel off your hair and scalp, skull. There's some liquid space in there, okay? Now we're getting down to the size of your brain. And your brain is probably the size, I always like to say, it's like if you could imagine a grapefruit with a pear stuck on the back. That's probably giving you a little extra, right? I would like to tell you it's the size of an orange and half of a pear, but I feel like some of you would be insulted by that. So I'm going to give you a grapefruit, right? Your brain's pretty small. Like, you can hold your brain like this, right? Like, that's your brain. It's probably all you got. It's all you need. I guess you made it this far. So, limited amount of real estate, right? If we gave every part of your body equal space on that little strip, because we're not even giving your body the whole brain, are we? We're giving your body like a strip this wide and this long, right, on either side. So we'll double that, right? We're giving you that much space. You've got to be very careful about which parts of your body you give a lot of space to. You don't want to give equal amount of space to everything. What's interesting about this is you would think, like, what's one way you could organize this? Well, in proportion to however big your body is, let's give that part of your body that much space in your brain. That makes a lot of sense, right? But that's wrong. It's the wrong way to do it. And the reason for this is, but don't say out loud, all of the things you think you can do, we're going to think about two body parts. We're going to think about a small body part, and we're going to think about a big body part, right? So for the small body part, I want you to think about your lips. Okay? I want you to think of all of the things you can do with your lips, and primarily all I really want you to think about is speaking and eating. Okay? Facial expressions. And we're going to leave it at those three things, right? You can list whatever else you want sort of mentally, but don't say that out loud. So those are the things you can do with your lips, right? That's the end of the list as far as we're concerned. What are the things you can do with your back? Nothing. There's nothing on the list, right? You can wear a cape. Like, that's the extent of what you can do with your back, right? So why would I give my back, which is maybe this, I'm just exaggerating, this big, why would I give it a lot of brain space? I wouldn't, right? The lips, think about all the fine movements you have to use to speak, right? That's probably going to require a lot of processing space, right? A lot of real estate. So you're going to put a lot of space for your lips, not so much for your back. Sort of similar comparisons, let's think about your legs, right? You can move your legs back and forth, that's awesome. Not a whole lot else. But think about your hands, right? Think about all of the, right now, how many of you are taking notes? How many of you have used your phone today? How many of you, I don't know, built a ship in a bottle, right? Whatever it is you do, your hands are very, very maneuverable, right? They do a lot of things. So they need a ton of space in your brain, even though they're quite a bit smaller than your legs, right? Your legs are going to get less space. Okay, so we're going to call that cortical magnification. Eventually I'll show you a picture of what's called the homunculus. Homunculus means distorted human, right? So if you ever want to, like, I don't know, it's a good insult to throw when you're, like, road rage situation. Get out of my way, you homunculus. Doesn't roll off the tongue quite as well as a few other things that you might say, but, work it into your vocabulary. We call it the homunculus because if you were to draw the body that is represented by your brain space, it's going to look weird, right? Giant hands, little itty bitty torso, right? Huge lips and big eyes. Ears are going to be actually pretty big too, but it's going to be kind of a weird structure, right? Because of the way we devote brain space. Does that make sense to everybody? This is going to be this concept of cortical magnification is going to work for all sensory systems. Like, some things are more important than others, right? When we get to the visual system, the part of the world that's like that few central degrees, right, that little bit in the middle, that's so much more important to you than anything in the periphery. Even though the periphery takes up so much more space, we're not going to give you that much brain space for it, right? We're going to give you just a little bit of brain space for that and a whole lot for what's in that middle, those few central degrees. Here we're doing the same thing. Hands are really important. You can move your hands a lot. You can feel a lot with your hands. They need a lot of space, right? They're going to take up a lot of brain space. They need a lot of brain cells to operate them. Your back, it's there, right? Doesn't really do anything. All right. So let's think about receptive fields a bit, right? This is fairly easy to think about in terms of thinking about your skin, right? But when we start thinking about receptive fields sort of out there in the world and we're not anchoring it to your body, it can get a little confusing, Alexa. So we just want to, let's anchor it somewhere first, right? And then later we'll let it float free into the world and think about that. Okay. Each one of your receptor cells and each one of your cells in your cortex, right, is only responsible for a small part of your body. You've got like 1 trillion cells in your brain, right? Okay. So why don't you let each cell kind of have its own little job, right? And when we think about your sensory cortex, we think about your somatosensory cortex. Each one of those cells is going to be responsible for only one small part of your body. Okay? one very, very small part of the surface of your body. And in fact, each cell is going to be responsible, because I know you remember that hypercolumn story that I told you, right? Each one of those cells is only really going to be responsible for like one type of thing that can happen in that one small part of your body, right? So you're going to have overlapping receptive fields for pain and temperature and tickle, right? So we can think about that. But if we think about a receptive field, now this is exaggerated because that's a huge sort of receptive field, and we'll think about why that is in just a few moments. And on Thursday, when you're randomly stabbing people, we're gonna do it for science, and we're gonna measure those stabs, okay? And we'll think about that in just a few moments. So you have these receptive fields. Typically, the structure is what we call center surround, right? And so if we were to take that receptive field and draw that out, we would see that the center of that is excitatory, and the surround is inhibitory. Because what's important is to know what's being activated, but also what's not being activated, right? You want some real clarity and definition to this, right? You want to know where the on is and where the off is, okay? And that's going to be sort of important to you. You can measure this activity when you stimulate. For this purpose, you can think about, like, you know, kind of stabbing or poking, you know, these different regions, and you can see how the cortex, this is kind of the response, right, of those brain regions, okay? What's nice about this is the way they've drawn this is this shows you that sensory receptor. It shows you when you're in the thalamus, when you're in the brainstem too, and then when you're up in that sensory cortex, that somatosensory cortex, right? This is a right hand, so we're going to be in the left cortex, right? We're going to be in the left cortex. We're going to record from that cell when you excite that or activate that center part. It's excitatory. Look at all those action potentials. You get those electrical signals. If you excite the surrounding part, you don't get any action potentials. You get a little rebound afterward, but don't worry about that. And if you excite both at the same time or activate both at the same time, they kind of equal out, right? So that's like giving someone $5 but taking $5 from them at the same time. Okay, are you going to get excited about that? And some guy's going to mug you, and he's like, "Give me $5, but here's $5 back for your trouble." You're like, "All right, I guess," and you just move on. Okay. Questions about receptive fields. Your body's covered with these, right? You can think about your body covered with these overlapping small circles, right? The entire surface of your body. Some of these circles, though, are bigger than others. Do you remember that time I talked to you about cortical magnification? And I said some parts of your body are more important than others, right? Same story here. 2-point acuity. I would pay attention to this. Laura wants you to pay attention to this, right? Because what's going to happen 2 days from now? You're going to try to tell people how to do this, and if they paid attention now, it's going to be a lot easier. They're already going to know what to do, right? So we're trying to save some trouble. Classic way to sort of figure this out, and actually this is a great way to sort of estimate how much of your brain space, relatively speaking, is devoted to a particular part of your body, right? Because the smaller the receptive fields for that particular part of your body, the more receptive fields you need to cover the whole surface, right? So let's imagine how many of you are aware of tarps? You guys know about tarps, right? Those plastic things that you use to cover stuff, right? Okay. Now let's imagine we're going to cover, I don't know, what's something you own at home. Somebody is, one of you, I don't know who, I can just imagine you're a chainsaw artist, right? You get these tree trunks and you carve chainsaws, but the rain's coming and you don't want it, you don't want your tree trunk to get wet. You're going to cover it with tarps, right? If I give you tarps that are 2 inches by 2 inches, how many of those tarps do you need to cover? Like a lot, right? Okay, so it takes a lot of time. It takes a lot of effort. But if I give you one tarp that's like 10 feet by 10 feet, well, you don't need any more tarps, right? You just throw that one tarp over it. Receptive fields are the same way. If you have small receptive fields, you need a lot of them to cover that part of your body. If your receptive fields are bigger, then you need fewer of them, right? If you have fewer receptive fields, that means you need fewer cells in your brain to get all of the information that you can from that part of your body, right? If you have smaller receptive fields, each one of those receptive fields needs a cell in your cortex, right? And so the more receptive fields you have, the more cortical cells you need devoted to that. Now, based on that story, which do you think has larger receptive fields, your back or your hands? Larger. Yeah, the back, right? Because the back takes up less space in the brain, all brain cells being roughly the same size, you need 10 brain cells to cover your back. You need 500 to do your hand, right? So you're going to have smaller receptive fields. You can measure this using calipers, a compass, a set of dividers. We're going to come in, I mean, you could do it with, you know, I don't know, 2 steak knives. Whatever it is you want to use, we'll probably do, we've got a number of, we'll use the compass, we've got dividers, we've got some other sort of drafting implements we'll bring in, and then if we really run short on that, sharpened pencils work perfectly. They kind of work the same way, right? What you do is you take this device and you poke someone and you go, hey, how many stabbers do you feel, right? And if they go 2, then guess what? Both of those are in separate receptive fields. Once you get them close enough that they go, I think you're only using 1, then you know both of those are inside that same receptive field, right? They're only activating that receptive field once. Because all your receptive field knows is, I'm excited or I'm not. It doesn't know I'm excited because 2 stabbers, it just knows I'm excited, right? And if you look at this nice chart, and you guys should get similar data, and we're not going to use all of the parts of your body that are listed here for some obvious reasons, right? Certain parts of these you're not going to, like, you know, hopefully not going to, like, you know, expose in class, right? So let's take an example, tip of the tongue or tip of the finger. Both of those are very, very sensitive regions, right? How many of you use the tips of your fingers for all kinds of amazing things, right? Like typing. How many of you have ever typed? Assuming most people have, right? That's all tactile, right? You don't even really look, right? You just know where the keys are. You can kind of feel the keys, right? How many of you know someone who's visually impaired and they use Braille? Or how many of you are just aware of Braille, right? That's again, tip of the finger. If you've ever seen Braille, those dots are really close together, right? So you have to have that very sensitive sort of thing, right? You're not using your back or your neck for Braille, right? Nobody's taking a book and like rubbing it on their back and like trying to read that. It doesn't work, right? The dots would have to be like this big, right? And you have like 2 dots on your back, and you're like, well, that was 2 dots. That's all you're going to tell. So what we'll do on Thursday, I don't want to, like, you know, ruin the surprise, Laura, but we're going to have you in your groups kind of measure this information multiple times so you can get good, reliable data. But we're going to use a couple different parts of your body. There are a few parts of your body that you can, you know, easily and willingly expose in class, right? Some good examples are your forearm, fairly sensitive, your fingers, obviously. Some folks are okay with the back of the neck. Some of you that might be more difficult. Back of the leg is kind of good. The calf, right? It's kind of a good spot you can do as well. Okay. So there you go. Or forehead, you know, you can just let people jab you in the face. Hopefully the other person's not too shaky, right? They miss and get you in the eye. All right. So based on this information, you can think about which receptive fields are larger or smaller, which parts of your brain or which parts of your body have a bigger representation in your brain. It's all connected, right? However you want to think about this. Questions about that? All right, great. Right. So when it comes to somatosensory perception, right, just a sort of sensory aspect, we want to think about sort of some different concepts here, right? Detection is one thing. Discrimination is what we just talked about, right? Every time someone stabs you, you're going to be able to detect that, right? They're going to do that. Hopefully not too much pressure, but enough pressure, right, so that you can detect that. But the discrimination is it 1 or is it 2, right? What's going to be amazing is they're going to stab you with 2, and at some point you're going to indicate that that's only 1, because that's all you're going to feel, right? So that's where that discrimination factor comes in, right? Which is really interesting. We could think about pain. One of the interesting things about pain is pain travels through some of the pathways as temperature. Okay. How many of you have ever felt something so hot or so cold it was painful? Yeah, there's a certain threshold where if you get far enough above or below a temperature range, you're going to feel that as pain. One of the reasons you feel pain is you're concerned about tissue damage, right? If you do something and it hurts, probably shouldn't do that again, right? Because that's inflicting some damage to your tissues. And it's the same thing with hot and cold. We also have texture perception, right, which is kind of interesting. There are sort of two things that we might think about, spatial and temporal, right? So if we were to just feel something, kind of put our hands on it static, that's the spatial. But if you move your hand across it, that's going to be temporal, right? Like as you're moving your hand, how quickly do you feel changes in that texture, right? This is actually how Braille works. They'll move their sort of fingers over the surface, and they can tell the changes in that texture over the time that they're moving their fingers. They can determine what information has been coded there. So there you go, right? Sometimes you need that, texture and that temporal information, right? I know you're all big fans of corduroy. Everybody is, right? You don't see a lot of corduroy these days, do you? Not down on anybody who is or will in the future wear corduroy, right? You think like my entire wardrobe now, I cannot wear to class because he made fun of corduroy. But if you think about corduroy, for example, if you just put your hand on that, sort of static, it's not really giving you a ton of information, right? You have to move your hand across that so you can feel that change in texture, that temporal sort of information. Temporal just means time. Don't want to throw you off too much. Okay. What about object recognition? We talked about that haptic perception just a bit, right? That's going to be, again, related to being able to feel things, right? Touch, and then kinesthesis, right? So you're going to be moving something around in your hands. Folks are pretty good at this, actually, right? How many of you have some dark container that you stick your hand in from time to time and try to pull out a particular implement? Anybody have a backpack or a purse, right? Yeah, so you stick your hand down in there and you're feeling around for, I don't know, that one pencil that has a particular grip that's full of lead, right? And how are you like determining that one from all the others, and then magically you pull that out, right? And you're like, whoa, look at that cool trick. That's all through haptic perception, right? So that's kind of interesting. I don't want to go into this too much today, but there is an idea that your brain, your sensory capabilities are divided sort of into two broad, determination pathways, right? So if you are having a sensory experience, sort of two broad things you want to figure out. What is it and where is it, right? Those are the two things you want to know, what and where. These are handled through, typically, this is a pretty good rule, through dorsal and ventral brain regions, right? Dorsal, if you think about, my great example is if you don't know what dorsal is, think about a dorsal fin. If you don't know what ventral is, think about a ventral fin. Probably doesn't help you any because there's still two fins. Doesn't really give you a lot of information. Dorsal's on the top, right? And ventral is not on the top, okay? Typically, we would say it's on the bottom. When you're thinking about your brain, you're like, it's already at the top. How can it be at the bottom? But it's the bottom of the brain, right? And so we have dorsal, and then we have ventral pathways that handle this information. We'll talk a whole lot more about that when we talk about other sensory systems, right? This dorsal-ventral division, but I want to briefly sort of introduce you to that. If you want to know what something is, if you want to recognize what it is, that's going to be a ventral pathway, right? It's kind of going through that bottom pathway. If you want to know where something is, that's going to be a dorsal brain region. This is going to be true for the auditory system. It's going to be true for the visual system, right? This rule is going to be true. This dorsal-ventral division is going to be an important thing to keep in mind. All right. Questions about those sort of cutaneous or proprioceptive sort of things, because we're going to shift just a bit and talk about the vestibular system, if you guys don't have any questions. Did you have any questions? You're sure? Okay, great. I just wanted to check. All right, your vestibular system. Most of the time, people don't think about this one, right? For most people, it works, and it works most of the time. There are a few examples where it doesn't work, right? If anybody is afflicted with vertigo, that's a situation where there's a problem, right? You can get vertigo for different reasons. If any of you are afflicted with alcohol, sometimes your vestibular system will not work properly, right? Those are sort of two examples of situations where that would happen. Interestingly, what alcohol does is it changes the density of the fluid in your inner ear, right? Briefly, and it can cause you some things move around faster than they should.
What causes vertigo? Because I've been having it for like the past 2 days every time I turn my head to the left.
Really?
Yeah.
Well, that's interesting. Wait about 6 slides, and we'll talk about that. We will discuss vertigo a bit. I would recommend, I mean, unless you know what the problem is, I'd go see somebody to figure out what the problem is, because there are a whole host of things that this could be, from you've got an earache to, or you got like some fluid in your ear, right? Something messing that up, to you got a brain tumor, right? And so there's like, you see why there's such a wide range there. I don't really want to like say like, oh, you're fine, let it go, but I also don't want to say like, run and have your head cut open to see what's in there. The vast majority of the time it's something much less than brain tumor, right? Okay, so you should rest a little easier on that, but if it doesn't go away soon, I would recommend seeing someone, right? There could be a lot of reasons why something's going on, so we'll talk about that though. And it may not in fact be, we'll talk about it in just a minute. Just hang in there. Don't rush out and make an appointment right now. This is important information. I think you're going to live until the end of class. Yeah. Good news, right? That's the good news. All right, so the vestibular system, you sort of have two sets of organs, or 2 organs that we want to think about, and then you have 2 sets of each of those, right? Because you've got one on the left and you've got one on the right, okay? You have your semicircular canals, okay? And that's these 3 canals. It's kind of difficult to see on this drawing, but they're organized in a way where they're like at 90-degree angles to each other. That way, no matter how you rotate your head, one of those is going to be in the plane that you're rotating, right? The other sort of organs that you have are these sacs. You got a couple sacs there that are going to be important for determining which way is down, right? Those are your gravity sensors, which are kind of exciting. In general, the way both of these organs work is they're full of something called endolymph, right? So there's like some gelatinous fluid in there, right, that kind of moves around. And you have these what we call otoliths, right? Where they're kind of like, call them ear stones, right? You've got these kind of like chunks of stuff in there that move through that endolymph, and they'll go by and they'll brush over the top of cells, right, that have these little cilia sticking up, and when they hit those and deflect them, they cause those electrical signals to occur. What's interesting about this is because you have sort of, you know, one set in each side of your head on either side of the pivot point. This is your pivot point, right? When you turn your head, when you rotate, one of those is going to be sort of giving you information in one direction and one in the other, and you can compare the outputs of both sets of your vestibular organs to determine which way you're rotating or which way's down. You have to kind of get information from both. If for some reason one of those is not working very well, you can sort of get some weird information. Sometimes folks who have inner ear problems, right, this is what we might think about if they've got something jammed in there or some other sort of situation, you'll notice they'll tilt their head a little bit, right? Have you ever kind of seen them walk? They'll walk with their heads tilted one way or the other. The reason for that is the information they're receiving from one set of vestibular organs doesn't match the other. It's kind of off, even though their head's level, and so they need to rotate until they're trying to get that equal information. So they think the world is flat again. And I don't mean flat in the like idiot sense. I mean flat in the, you know, like not at an angle sense. Did that make sense? Anybody who is a flat earther is an idiot. I'm just gonna like, and if any of you are in that, I'll tell you to your face. You can call your mom. I'll tell her too. It's just the way it works, right? It's a big sphere. It's kind of a lumpy sphere, but it's still a big one. Okay. Questions about that in general? We'll do some fun stuff with this. These are the different ways that your head can rotate, right? We've got, anybody here fly airplanes? Anybody have the Microsoft Flight Simulator? Nobody's interested in that? Okay. But I'd check. Sometimes they use some weird words, right? Like roll, pitch, and yaw. So if you've ever kind of heard those words, it's just the three different planes that you can kind of rotate around. If you think about your semicircular canals and the way that they're oriented, you can see now those sort of three 90-degree planes, right? All right. Here's sort of a close-up view, hopefully a little better of those canals, the utricle and the saccule. Later we'll talk about this guy called the cochlea when we talk about the auditory system, but don't worry about that right now. Okay, we can kind of let that go. When you sort of accelerate your head, right, in some direction or another, okay, or if there's like a tilt and you just kind of hold your head there for a while, right, these otoliths, they move, as we discussed, and they'll deflect the cilia. These cells have little hairs that stick up like this, and they can move in one direction or the other. All right. So, different kinds of acceleration we want to think about. We think about rotational acceleration. Anybody in the gymnastics or accidentally rolling down a hill? Right? Happens all the time. I'm going to assume you've done one of the two, right? yeah. Which is that? None of you are, I don't see any Mary Lou Rettons out there. So I'm going to assume most of you have fallen down a hill and rolled, right? So you can kind of think about this rotational acceleration.
Well, you kind of have to do both with gymnastics.
You do.
You do a back roll. They start you on a hill. Down.
There you go, right? They roll you down a hill. Seems like a job I'd like to have. It's like, do they put you in a barrel?
No.
Oh, that's too bad. Because I want to put people in barrels and roll them.
The worst thing I did was they played like this weird opera song and they made me do like a split and they made me do it through the entire duration of the song and then I'd have to switch my split and then they'd restart the song. And
switch your split. How's that work? That seems like a problem.
They wanted me to like hyperextend it for like tumbling and stuff. So they were like, all right, we're going to hyperextend it. So by that they were like making me hold it longer and then like putting it on top of things. It was awful.
Sounds painful.
Wonderful. Yeah, I had to hold it for like 5 minutes each. So
Right.
you could do a right one, and then a left one, and then a middle one, and sometimes the instructor would stand on you and push you down.
Wow. You thought it was bad I was rolling people down a hill in a barrel. That sounds really bad. So, rotational acceleration, though, it's going to cause fluid to move in these canals, and then those receptors are going to respond as the otoliths move. Again, we've got those three canals, right angles to each other. Okay. When we're thinking about linear acceleration, right, you're not rotating. Now you're just deciding to like run really fast forward, or I don't know, maybe you're taking off in a spaceship, whatever it is you do with your life. That's going to cause a different sort of situation where we're not getting these differential, right, sort of rotations. We're getting it both in the same direction, right? And we can kind of figure out what's going on there. Okay. And this is where we're going to have the two vestibular sacs are going to get involved. They're actually oriented in a way that they can detect vertical and horizontal, right? And so that way any kind of tilt that you have, you're going to be able to determine that that's going on, okay? The cool thing about this is they always work down. They'll always tell you where down is, right? The main sort of underlying issue with vertigo is, and any of the, eventually we're going to talk about some vestibular illusions. You don't typically think about those, but they're the best. They're the ones that'll make you vomit. The vestibular system is giving you one set of information, and your visual system is giving you a different set of information. And they're both telling you down is in a different direction. And when they tell you down's in a different direction, it can cause you some serious problems, right? And that's what you get with vertigo. And then there's height vertigo, which is slightly different, but you have to be on top of a building. So that's fun, right? And then you can spin around in a chair. We'll all do that sometime. It'd be a fun lab. That's what we should have done for the lab for this lecture, is just spin people in a chair and see what happens.
That would be fun.
Yeah. They don't give me barrels. They do give me chairs with wheels. That was a mistake. All right. Your vestibular pathway, there's like 4 major pathways for vestibular information. 4 major targets, right? And so we're going to think about where some of those are. One is the cerebellum. The cerebellum is this like broccoli cauliflower thing that's at the back. Remember that half of a pear I told you had back there? Okay, well, instead of half a pear, imagine it's like part of a broccoli crown stuck back there. It's really what it looks like more than anything. This guy is important for movement, right? So it gives you information, particularly feedback about what you've done, right? So if you took a step and you hit the ground with your foot, that's awesome. You expected that, right? So you're going to match that up with your cerebellum. If you take a step and you don't hit the ground, that's a problem, right? And so your cerebellum's going to go, hey, wait a minute. Where did the ground go? Okay. How many of you have ever sort of stepped through the ground or the floor, right? And you've, like, there were stairs or there was a hole and you didn't realize it. You caught yourself sometimes, not all the time. You try to catch yourself. That's your cerebellum kicking in gear going, wait a minute, I expected a hard surface. I expected some feedback. I didn't get that. You can imagine why your cerebellum wants information about which way's down, right? Because if it's helping you with these coordinated movements, it's important to get that information in there. Okay. The other way, one of the other pathways is the thalamus. Again, this is just a general sensory sort of input pathway. This is the place we've talked about so many times. We've already said a lot of sensory information goes into the thalamus. Because all the other sensory information is coming in there, again, it also wants to know if you're moving or which way's down. We'll send some information to the spinal cord. Okay, we have this vestibulospinal pathway. There are a number of reflexes that we'll think about, some other sort of postural issues that we'll talk about. And finally, the vestibulo-ocular pathway, right? This is what helps you sort of stabilize your gaze, right? Your gaze direction while your head's moving. How many of you have ever, it's kind of a fun thing, you look forward and you move your head, right? And if you continue to focus on the same spot, you notice your eyes move, right, to stay focused on that spot. How many of you have ever walked across campus and you've been looking at something in the distance, right? You stay focused on that, but your head is moving, right, as you're moving up and down. As you're walking, right, your head kind of moves a little bit. You don't have to really work on that, right? You don't have to think like, oh, I got to move my eyes back down or up as I'm moving up and down on campus or across the ground, right, as you're moving. Because this vestibulo-ocular pathway, you know, as those changes occur, you have that information coming in. Okay. Here's a nice diagram showing you those pathways. This is maybe even more confusing than the other pathway that I showed you. I'm not going to ask you to memorize it, but these are your vestibular organs. You've got information coming and going back and forth, a lot of good feedback stuff. Again, a lot of sort of postural things going on. We won't think much about vestibular cortex, but there it is. And then stuff going over to your eyes. Okay. I do want to spend some time, though, talking about vestibular perception. There are some interesting perceptual effects associated with the vestibular system. Some of these we can induce, right, at any given moment. Some of them happen to you sort of given different situations. All right. So in general, what is vertigo? Again, as we've said, vertigo is just you have some difficulty figuring out which way is what we call gravitational vertical, right? Which way is gravity going? In case you don't know, gravity always goes down. If you want to know which way is down, that's like toward the center of the earth, right? That's the most important gravity force on you at any given moment from an individual level, right? There's obviously, you guys understand how gravity works, right? You just drop something, you can figure it out. Okay. When you get a mismatch in this downward sort of information compared to some of your other primary senses, in particular to vision, right? That's when you have a mismatch, okay? And that's when you can have what's called vertigo. It'll cause dizziness, nausea, vomiting. You can lose color, right, in your face, and you have some postural instability, right? And so this can be pretty severe that this happens to you. There are, again, a number of things that can cause this. In particular, we want to think about issues with your inner ear, right? We want to think about issues with those vestibular organs, something changing the way that they're working, some extra pressure in there, right, that can cause problems. If you have an ear infection, if you've got something in your ear that can cause you some problems. Anybody had an ear infection before? I think most people have. It's pretty standard, right? You may or may not remember, but you probably had some sort of issues with vertigo. So that's fun, right? Height vertigo is very similar. It's typically related to being up on a ledge, right? We'll talk at some point about how when you are high up on something and you're looking out, there's some distortion in the way that information is received, that visual information, right? You're going to perceive things maybe as closer or farther away than they actually are at that time, right? And so that, again, we're talking about a mismatch between, in this case, the visual system and your vestibular system. It's going to cause you some problems, okay? So there's height vertigo. Both of those are, things to think about. I'm trying to decide right now if I want to tell you a story about a sloth or if I want to wait. I'll wait just a moment. Motion sickness. Yeah, motion sickness, right? What happens here is one of your senses tells you nothing is moving and the other one tells you something is moving, right? And that can cause you some problems again, right? And so you can have some issues there. Vestibular system says one thing, visual system says another, which is kind of an interesting situation. What about postural alcohol nystagmus? That's a long name. And basically what it means is if you get drunk and you lie down, your eyes will move. That's exactly what it means. I wouldn't recommend getting drunk just to see this happen, but I'm certain you're going to be around someone at some point in the future who's had too much to drink, right, Alexis? And as a good friend, you're going to want to make sure that they're not up moving about. Then they can cause problems. So lie them down on their side on the couch and watch what happens. And in fact, you'll get these small eye movements called nystagmus. So the eyes will sort of like go over to one side, and then they'll just kind of like vibrate there, right? They'll kind of bounce on one side. I had a student once who had a brain injury, and he had nystagmus. So from time to time, his eyes would just, bounce over, go from there. He was in a motorcycle accident, I think, and had a head injury. So those are things to think about there, right? And nystagmus happens, right? So just because your eyes do that doesn't mean you're necessarily intoxicated. This will happen with, they usually do it when, we'll test it at some point later in the semester when we talk about the visual system. You can do it with like light and dark bars. So if you're kind of like rotating bars and you watch that spinning from time to time, your eyes will try to track that, and then they'll go back and reset. It's kind of a really fascinating thing. What about the oculogyral illusion? That sounds like fun, right? Okay. This is when you spin around in a chair and then all of a sudden you stop and the world seems to keep moving but in the opposite direction, right? Have you ever noticed that? You can do it now. If you want to take a moment, go ahead. Spin yourself around, 15, 20, 30, 40, 50 minutes, I don't care. And then stop. And if you're rotating in one direction, when you stop, the world will seem to kind of move back in the other direction, right? The reason this happens is you've stopped spinning, but guess what hasn't stopped moving? The fluid in your semicircular canals, right? So this is kind of like, if you've ever, if someone's ever slid maybe a mug of what's your favorite beverage, milk, at you, right, across the top of a counter, maybe a hot bowl of soup. You stopped it, guess what happened? The bowl or the mug stopped, but not the liquid inside of it. The liquid kept moving, right? And so that's what happens here. The bowl stopped, your head stopped, but the liquid, the endolymph, kept moving, and so you're getting that information from your vestibular system like, I'm still spinning, but your visual system is saying, no, everything's stationary, and so to rectify that, you sort of get this backward motion. We have the oculogravic illusion, right? That's kind of an exciting one. Okay. This is when you undergo linear acceleration and you feel that your body is tilting, right? So this can happen if you're like in a car and you start going really fast. You might feel like you're tilting, right? But you're not. Okay. The reason for that is, again, that acceleration is moving the otolithic membrane, right? Those otoliths relative to you. And when that normally happens, you're tilting, right? But in this case, you're not. You just get that sort of effect. This is a cool one, actually. Vection, right? We should talk about vection. This is when you think you're moving, but you're not, and your surroundings are actually moving. This is, I don't know, a lot of you don't ride trains anymore, right? I don't know, I'd recommend a train ride once in a while just to see what it's like. But if you are sitting maybe at a stoplight, and the vehicle next to you starts slowly moving forward, and you'll feel like you're going backwards, right, Aspen? And it happens. You're not moving, but because it's moving away from you, you're getting that sort of mismatch of information, right? So your visual system, because of the way your view is sort of windowed around that, your visual system is saying, there's movement in my world. It's global motion. We'll talk about what that is later, right? But your vestibular system is saying, no, I'm not going anywhere at all, right? So you kind of get that, again, that mismatch, and you get that interesting sort of situation. This can happen either linearly, which is typically what we would expect, or it can actually happen in rotation. That's called circular vection. It's a really fancy name, right? Finally, Coriolis effect. This occurs when you get in the, you're like rotating, but you tilt your head at the same time, right? And because you're doing that, everything's sort of getting screwed up, right? All of your visual information and all that vestibular information is getting crisscrossed, and you can kind of have a little confusion about what's going on. Okay, so those are your sort of vestibular illusions, right? If you want to kind of go home and practice these, you're welcome to do so. I would like to talk to you briefly about sloths, right? And this is just it's that moment in the day when you're like, let's get a sloth story. What's really fascinating about sloths is they're slothful, right? They don't move very fast, okay? They're quite slow. If you think about humans, for an example, we're actually quite fast, right? We move around quite a bit. Despite the fact we're not in trees anymore, you would have thought something in a tree would be more acrobatic than humans, right? But sloths don't fall into that category. They're not more acrobatic than humans, right? Humans are more acrobatic. What's really important to a human is having a good set of vestibular organs, right? Can you imagine trying to get through your life if your vestibular organs were screwed up? Think about your experiences now and how distorted that is with just like a small disruption, right? But what if it was a much more profound disruption? And so if you guys would let me, I could cut open all your heads and remove your vestibular organs, and they would all be nearly identical, right? There's very, very little variation in the DNA that codes for your vestibular organs and the way that those vestibular organs are structured. Okay, there's very, very little difference from person to person. Sloths, on the other hand, are a completely different story. There's actually a lot of variation in the DNA that codes for vestibular organs, and there's actually, as far as we can tell, a good bit of variation in the actual structure of their vestibular organs, right? And if you think about this, what happens if a sloth falls out of a tree? It's dead because something's eaten it, right? Doesn't have time to get back up. Vestibular organs are not that important to a sloth because they're not moving fast enough either in a linear or a rotational sense, right? For good vestibular organs, or rather bad vestibular organs, to have actually been an issue, right? If a human has bad vestibular organs, like really bad vestibular organs, guess what happens? You can't figure out how to get out of bed in the morning to go have sex with somebody and make a half copy of yourself. So those bad semicircular canals never make it to anybody else. That's a good thing, right? That's how we all got here. Think about that, right? All of our ancestors had really good vestibular sacs. It's not something you think about. The sloth, on the other hand, because the sloth's not moving fast, it doesn't need good vestibular sacs. It doesn't need good semicircular canals, right? And so if it has a bad set, it's not any less attractive to another sloth than, or any less likely to meet up with another sloth than it was if it had a good set of vestibular sacs, right? So it's not going to be limiting to its reproductive capacity in the same way that it would a human or some other species that would require, some sort of important information about the way it's moving through its environment. See, that was a good story, wasn't it? I knew you guys would enjoy it. That's not in your book. It's not surprising it's not in your book, right? It should be. I think it should be. If they ever ask me what would make the book better, I was like, more sloth stories. Let's do that. But I think that's an important story. It's an important thing for you to think about. I do like to try to highlight when there are differences between species, right? We'll talk more about later, bats, right, and how they rely more on echolocation than they would vision. So they're auditory cortex is expanded relative to humans, right? And so sloths, they probably have a very small vestibular cortex. You know, humans have a vestibular cortex. If you think about spider monkeys, they probably have a rather large vestibular cortex, right? They're constantly, spinning and doing things, right? But if that species is not relying on that as much, you don't want to devote that brain space to it, right? Remember, your brain is very small, right? And so you want to be careful what you put in there, right? You want to be careful what you devote that real estate to, if that makes sense. Questions about that? Great. So here's some points you should remember. I'm assuming you're going to forget some of those, so we'll just skip past that. For Thursday, right? Let's just think a little bit about what we're going to do on Thursday when we come in and get started. Come in, get in your groups, because we do want to make sure that we collect data from multiple people, right? Because we're going to look at averages and so forth, right? It's not just going to be like, oh, here was the distance, right? It's going to be important on Thursday when you do the testing, whether you're the